Assalamu alaikum. A very warm welcome to another discussion on our series on CLI, Clinical Laboratory Instrumentation. What we're interested in today is basically HPLC, High Performance Liquid Chromatography, and it's sometimes also called as High Pressure Liquid Chromatography. We'll see why it's called that in just a moment, but before we get to that, let's just first understand the underlying motivation for it. Why are we so keen to study this? We know that chromatography is a separation technique, of course, but one of the major applications for this kind of chemistry is usually testing and analysis of suspected illegal drugs, for example, in body fluids amongst athletes. And then there's always a research aspect to it as well. So basically in forensic science, this is a very important analytical tool. So let's just quickly revisit some of the fundamentals of chromatography now. And once we're done with that, we'll briefly develop an understanding of TLC, the thin layer chromatography. And then we'll look into how GC, the gas chromatography, works. And then we'll finally try to understand HPLC, high performance liquid chromatography. So the best place to start is simple paper chromatography. Try to understand the basic concepts that come at play over there. And before we get to that, we'll see a quick demonstration of a video in which we're seeing, it should appear on your right hand side now. Yes, there it is. It should basically show capillary action at play. All right. What we see here is a prime example of capillary action, which is basically just the ascension or the sucking up or the travel of liquids through a tube or cylinder or permeable substance. In our case, it's just a tissue. It could also be seen in a straw. So it happens because of the adhesive and cohesive forces interacting between the liquid and the surface. In our case, the liquid is water and the surface, and that I'm referring to is tissue. So sum that up, basically capillarity occurs when intermolecular bonding of the liquid is weak, it is inferior as compared to the bonding between the liquid and the surface. So when the bonding between the molecules of the liquid is weak and the bonding between the liquid and the tissue or the surface is strong, that's when we get to observe this phenomena of capillarity or capillary action. So we refer to this intermolecular bonding of the liquid, the mutual attractiveness which maintains a certain shape of the liquid, as cohesive forces. So we call them as cohesive forces, whereas the attraction between the molecules of two different surfaces, in this case the water and the tissue, the attraction between these two different surfaces, that's something that we refer to as adhesive forces. So there are two forces at play, the cohesive forces and the adhesive forces. Again, capillary action occurs when these adhesive forces are stronger than the cohesive forces. A good way to memorize this or remember this would be the adhesive starts with AD, refers to when an additional or an added material is involved, and cohesion or cohesive is at play when the forces between the molecules cooperate together, when their mutual interaction is strong. So AD and CO would probably be a nice way to remember this. So there are many different types of chromatography and the techniques associated with them, but they primarily follow a very basic or at least similar principle. What we've called as the just basically we've called it the tissue or called it the surface, but what's basically there are two important things in chromatography when we talk about them. So what's called a stationary phase is the absorbent material. It can be paper, it can be tissue, it can be silica, but that's the stationary phase. And then there is a mobile phase, which is either a liquid or a gas, or it basically flows through the stationary phase. So the components in the mixture flow through it as well, and then they tend to separate out. So basically we are interested in these two things when we talk about chromatography. The stationary phase, it's the material, it's stationary, it's not moving, it's just right there. And then there's a mobile phase that passes through this stationary phase. So the mobile phase is basically what contains, it can contain our sample, or our sample could be placed such that the mobile phase goes through it and then carries it with it. So you could tell intuitively now that if you run your experiment for a long time, as time goes by, the components, they get to the end of the stationary phase. And the simplest and easiest and the cheapest way or technique that this is done is TLC, which is the thin layer chromatography. The stationary phase in TLC is a thin absorbent layer of silica. It's typically silica, it could be alumina, and it's coated on inert backing plate. So, which is typically a glass or plastic, could be aluminum. As you can see in the video, the equipment is rather simplistic. It's not a complicated equipment. It's very simple, which is what makes it very cheap as well. And it's pretty much doable in laboratory or simple environments. So total distance, however, that the solvent, our mobile phase, is going to travel is called the solvent front. And the ratio that is traveled by a component in the mixture, that ratio of this distance traveled by the dye to the solvent front is known as the retention factor. It's basically just the ratio, and this ratio is obviously different for different components in the mixture, and based on the distance that different components travel, this retention factor turns out to be different for different components, and that is how we identify different components. So sometimes these spots, they don't quite appear to the naked eye, and that's because they could be colorless. And for a range of techniques, there are a bunch of techniques. There are a range of techniques that could be used to visualize them. We could put a TLC plate under a UV light and apply chemical reagents to make them appear. One such example is on your right, on my right to your left. But there are different techniques, however, that we could employ to make sure that our components are visible, and we could look at them. So it's important to understand that a TLC plate is only just a few centimeters long, and then there are millions of organic compounds. So it is possible that their spots travel similar distances, and it becomes quite difficult to distinguish different dyes or different compounds, as they could appear very close together. So and that could be a problem in itself. So TLC serves, although as a quick, cheap, simple way to identify the presence of compounds, but it's not a concrete way to identify these compounds. So it's not a very reliable way to identify them, as certain spots could appear together. Their distances are not very the resolution is not very nice. You could see these dyes appearing together and then find it rather difficult to distinguish between them. The next up is the gas chromatography. So the gas chromatography, on the other hand, does not use a plate. As it's evident from the name, the mobile phase in gas chromatography is a gas, but let's talk about the structure a bit. It uses a metal tube. So that metal tube houses or packs the stationary phase. And the metal tube is wrapped around a coil inside the instrument, and the material, the mobile phase, which is fed from one end and allowed to pass through the stationary phase. So the mobile phase is often mixed up with the sample, and then it's allowed to pass through the stationary phase, and then it's detected at the other end of the coil. So intuitively, you could understand the separation of the components in gas chromatography is based on how much time a component takes to come out of the other end of the stationary phase, of the other end of the coil. So a fundamental difference between TLC and gas chromatography GC is that, of course, the mobile phase is a gas, but the gas chromatography is keeping track of components based on the time they take, whereas the stationary phase, the time they take to pass through the stationary phase, whereas for TLC, it's about the distance as opposed to the time that it takes for the components to travel. So gas chromatography is time-based component identification, and TLC is a distance-based component identification. So while we were finding a ratio between the distance traveled by the dye to the distance traveled by the solvent, and we called it as a retention factor, in gas chromatography we are interested in the retention time, which is time traveled by time it took for the gas to travel through the stationary phase or through the coil. So we are more interested in the times when it comes to gas chromatography. The received components then could be when they're detected at the detector in gas chromatography, they're plotted and then they're compared with known standards for identification. It's very much similar to what we would do in a paper or a simple TLC. You would compare the dyes. If you have time, if you're keeping track of times, such as in gas chromatography, you would compare those times with standards or already known samples, and then you would make a decision of what's in there. So TLC is when it comes to basically which technique should you be using, it depends on your compound, it depends on the mixture that you've got, because as you'll see in the video as well, there's an oven and there's a lot of heat that's involved in the setup as well. So from substance to substance, it greatly affects whether or not they could withstand those temperatures. And one of the things about GC is that although you get these nice peaks, but the resolution is not so nice, which means that similar or times that are very close together for different components, they may tend to overlap, so it could become rather hard to identify exactly which component is present by just looking at the peaks, because some components could have very similar retention times. So there's a drawback with the gas chromatography when it comes to that. However, the advantage of gas chromatography, though, is you could quantify exactly how much component was present in that mixture. Because you have those peaks and all of that component has passed through the coil, you could integrate that peak, find out the area under that peak, and quantify exactly how much component was there in the mixture. And this is very much similar to what we did in spectroscopy as well, in NMR spectroscopy, when we were quantifying the hydrogen atoms in different molecules. I think we looked at a lipid molecule and a lipid signal, I think from one of the examples, and we were trying to quantify how much is it, and that was about computing fat fraction. But anyways, from here on we could look into the amount of component present in the mixture, and that is what GC could give us, but the TLC cannot, because you cannot just look at the dye, color of it in a thin layer chromatography and judge from that dye that how much quantity of a particular component was present in the original mixture. So a thin layer chromatography would not give you the quantity, but the gas chromatography does. So that being said, let's move on to HPLC. Now, high performance liquid chromatography is very much similar to gas chromatography, but you'd have guessed it by now, the mobile phase is a liquid and it's not a gas. So that's a very fundamental difference between the two. However, long metal tubes are not used to house the stationary phase that we saw in gas chromatography. So instead of those long metal tubes and coil-like structures, we instead use steel columns or steel chambers or steel containers, and we use these because of the high pressure that's being used in this, so that these columns could withstand those high pressures. So to generate those high pressures, obviously we would require a pump, and that would we would feed in the mobile phase at a very high pressure. The question here is, why are we using a high pressure in the first place? To what advantage, basically? So that's a bit of a question. So since stationary phase is so densely packed in the container, it's a bit of an issue with the gas chromatography as well. That's what the issue is, that the stationary phase is so densely packed in the container that to get the liquid to pass through it at a useful rate, at the realistic rate, we apply a very high pressure, so that we don't have to wait for eternity for our components to separate out, and we don't have to wait for so long for all the components to come out. We instead use a high pressure pump, and that enables the liquid to pass through pretty fast and at a good flow rate, basically. So since the separation is based on how soon the component is detected, we keep track of the time again, and we also refer to this as tension time, very much similar to what the gas chromatography did. The peaks appearing on the chromatogram are similar, and they could also be integrated in this case as well to quantify the amount of component that's present in the mixture. So, but one underlying difference between the two is that the peaks in the chromatogram of an HPLC are at a higher resolution as compared to gas chromatography. So that greatly helps in identification of the compounds. To summarize that up, gas chromatography is relatively simpler than high performance liquid chromatography, and that is what makes it less expensive, and it's mostly faster as well. But HPLC, on the other hand, is more suitable for samples that are thermally unstable. You wouldn't want to put a thermally unstable sample in a gas chromatography chamber. As you saw, there was a heat oven there as well. So it varies basically on the nature of this sample, what you're trying to separate, and the amount and the resolution that you would need to successfully separate it. So based on different nature of the samples, you choose your technique accordingly.